Hey bio team, I wanted to do a quick refresher on how proteins get synthesized. So this is a close-up view of the inside of a cell. This line right here represents the nuclear membrane. So this half of the screen here is the nucleus. This half of the screen over here represents the cytoplasm. And so you can see inside of our nucleus we have a double-stranded DNA. And in the lower right-hand corner, we have a ribosome, which is made up of strands of ribosomal RNA. So let's say we wanted to use the ribosome to build a protein out of this DNA. Well, the first thing we'd have to do is the process of transcription. That is, an enzyme would have to unzip this DNA and transcribe a strand of it into a messenger RNA. So let's watch that happen here. We'll bring up the enzyme Rna Polymerase. And we'll watch it as it unzips the DNA and transcribe RNA in its wake. So after the messenger RNA gets made, it goes through processing before it leaves the nucleus. And so we'll watch that happen here. Now the first part of this processing involves taking out these useless nucleotides called introns. And these are left behind in the nucleus. Whereas the rest of the RNA that is going to be heading out of the nucleus, the exons, are stitched together. And last, before the messenger RNA leaves the nucleus, there is a guanine triphosphate cap added to the 5' end of RNA. Which will allow the ribosome to accept the mRNA. And there is a tail added onto the 3' end of the mRNA made of adenine. And so this is often abbreviated to the poly-A tail. Now at this point our messenger RNA has been processed to the point where it can head out to the ribosome. And at this point translation can occur. So the 5' G cap attaches to the ribosome. And at this point, transfer RNA, that's these guys up here, will start attaching amino acids onto the protein. So we'll watch that happen here. And what you'll notice is that each transfer RNA has a group of 3 nucleotides called anticodons, which are matching with codons on the mRNA. The other thing you might notice if you start looking at enough chains of amino acids is that the first amino acid in each protein is always a methionine, which provides the starting point for the chain of amino acids, which will eventually fold into our protein. And then it's not shown here, but the last codon that the ribosome reads before ending the protein is always a stop codon. And there's a couple of those, but they cause the ribosome to stop adding amino acids and to release the protein so it can fold up and go start to function. And that's it. At this point, you guys have some practice problems. We'll see you in class next time.